Hi, I'm Aditya, and this is me and Carlos's video presentation. This past summer, we joined the health research program under Dr. Kshitij's lab. Our topic was cancer research, primarily focusing on microscopic cellular invasion of cancer cells, on stromal cells, and cell-cell interaction, and cell tracking, and much more. And our goal was to gather enough data in different conditions and understand the relationship between stromal cells and cancer cells and how we can prevent a less invasive metastatic state. We were both new to the field, and so we were given a lot of tasks to better our initial understanding. Some of those tasks included linearization of articles, where we would be given a few articles and we were supposed to find and extract information on topics that we would be researching in. Another thing we did was research specific genes that matched with our research, finding their function, how they're associated with which cancer, and where can they be found. So for methods on our data analysis, we used an imaging software called ImageA to analyze microscopic images taken from the lab over certain time periods and gather data for each of our projects. For example, one of our projects was immigration and migration of cancer cells, and there we were given microscopic images of different locations and conditions under 36 hours of observation, and we had to record the 0, 12, 24, and 36 hour time points and gather the data of the area of invasion over those time points starting from 0 to 36. We use this software for majority of our projects. Here's an example of some data collection from one of our projects. On the left, there's 4 areas drawn in different time points, and each tracing is the area of a different time point. And on the right, you can see this is some data from our breast cancer cell line project, where we're calculating the area, the intense density of the cell, and the area of digestion of the cell. Some of the things we learned this past summer and continuing our research, we learned how research is conducted at a medical center of a university. We learned how to use other studies as a guide to extract data and better your understanding for your own research. We learned how to use imaging software to collect many different types of data. We learned how to analyze the data we collected and understand our results. And finally, we learned how that even though we were remote, we were still able to contribute to research conducted in a lab and come up with some meaningful data. For future goals, we hope to continue our research in this field and expand on what we've already done in order to hopefully maybe publish a paper with our promising results. The knowledge we've learned from this program is very valuable to both of us, both me and Carlos, because we both hope to enter the medical field, whether that's in a lab or in a hospital. And understanding how a lab works is a very important aspect of the medical field from a different perspective than a hospital would give you. And so what we have experienced here is very valuable to both of us. Finally, we'd like to take this moment to thank Dr. Kshitij for letting us work in his lab and be part of his research, as well as our PhD mentor, Ashkan. And we would also like to thank everyone that ran the health research program, because they were very helpful and built a sense of community between all the students. And we hope to continue this bond with both Dr. K and the health research program, and in the future grow on the research that we conducted this past summer and semester.